This conference will now be recorded.
Okay, thank you. Good evening, everyone. So we'll continue where we have left yesterday. Okay, am I audible? Connection was lost. Huh? Okay. So yesterday we dealt with what is acetylcholine, what are the ligands, what. So today what we will do, we will complete this muscarinic receptor. What are these muscarinic receptors? They are acetylcholine receptors, or cholinergic receptors. Okay, there are two types of receptors, nicotinic and muscarinic. So nicotinic we have already done yesterday. So we will finish this muscarinic receptor. Now, what is the structural form of this nicotinic receptor? Can you tell me? What was the structural form? How many subunits were present for this nicotinic receptors? Nicotinic receptors. It was 5. Yes, right. It was 5. Yes, right. Very good. And yes, pentameric structure. And what is the binding site for this acetylcholine? Yes, right. It is alpha. So, right. Yes. So it was alpha unit. Now, if we come to this muscarinic receptor, it is a 7 membrane spanning domain. Okay, 7 membrane spanning domain. But you will see here that here it is written M1, M2, M3, M4, M5. This is actually the subtypes. How many muscarinic receptor subtypes are present? They are of different configuration. Different on the basis of the location, their subtype has been categorized. So every M1, M2, M3, M4, M5, they all are in 7 membrane spanning domain only. But on the basis of their location where this muscarinic receptor is present, they have been categorized. Now, this was just for your information, because sometimes in exam, what they will do now, they will to confuse the question, they will give you this M3, M4 like this, the name of the receptor will be given. So you don't have to be puzzled at that time. You just have to take it as a muscarinic receptor only, whatever is the subtype. Okay, and it is a 7 spanning membrane domain only. So you can just go through this slide that where are they present, where are they primarily found out, but this is not that important. I think my internet is slow. It's not going forward. Okay. Now, very important thing is that we have discussed it in our first slide, that nicotinic receptor was a ionic receptor because it was opening the ion channels. That was the Na plus and K plus channel. But on the opposite of this, the muscarinic receptor, they are actually binding to some internal protein, And what is that protein called? That protein is called as the G protein. Okay, that is why muscarinic receptors are called as GPCR. That means G protein coupled receptor, because this is a receptor. Okay, this is a receptor and it is coupled to a protein which is called as a G protein. That is why it is called as a GPCR. Okay, GPCR. And you should remember that all the GPCRs are 7 membrane spanning domains. See the next line. It is a 7 polypeptide and it is made up of 20 to 25 amino acid, and when they are spanning this hydrophobic domain, they are doing it in this arrangement, which is called as an alpha helix. Now, one question. If you are attending the lecture from the first day, you will be able to answer this question. Do all these membrane spanning domains are alpha helix? All the membrane spanning domains are alpha helix. True or false? False, right? It is false, because very good, right? Majority of the membrane spanning domains are alpha helix, but not all. Okay, you should mark these words, that all and majority, they have a difference, because sometimes now they will ask you this types of questions. Okay, so all are not alpha helix, because there are certain amount of membrane spanning domains which are also in the beta forms. Now, what are these alpha and beta forms? When we are doing, when you are doing this biochemistry portion, proteins, you will get to know that what is this alpha helix and beta forms. In very short, they are actually secondary structures. Okay, they are secondary structures of your protein. Once your protein is synthesized, it is just a single chain of amino acids. But to get its structural and functional role, it has to be molded. So the second form is the alpha helix and the beta sheets. Okay, so this is an alpha helix structure. They are like this, alpha helix. Okay, they are like this. So in the membranes, they will be present like this. They will be spanning like this. And you should very clearly know that this is the plasma membrane, which is hydrophobic. Okay, hydrophobic. So what will be the nature of this alpha amino acids which are present here? What will be the nature of this? Will they be hydrophilic or hydrophobic? Yes, like will attract like. Okay, it is not like that, huh? In magnet, it is like and unlike poles, but here it is different. Okay, only phobic and phobic will have attachment. Fine. So if it is a hydrophobic area, the amino acids which will be present here on the outer surfaces, huh? On the outer surfaces, they have to be hydrophobic amino acid. Only then this structure will be stable. Fine. So this structure, it is made up of around 20 to 25 amino acid structure. Okay. One more thing you have to remember is that nicotinic receptor, nicotinic one, is always excitatory. It will always fire a neuron. Always. Okay, nicotinic can not be going into a inhibitory function. But but this one, muscarinic one, it can have both the function, excitatory and inhibitory. It can have both the function at these particular locations. The locations are not that important, but this is important, that the muscarinic receptor has both types of function, excitatory and inhibitory. It can give a sympathetic response and a parasympathetic response. Both at both the places it can have its role. So let's see that how is its structure present, and what is its role. What are these things? So this point is clear now, that they are G protein coupled receptor. What does this term means, G protein coupled receptors? It is linked to a protein. Okay, it is linked to a protein which is called as a G protein. That's it. Now see here. This is your muscarine. Why it is called a muscarinic receptor? Even though it is binding to acetylcholine, but why it is called as a muscarinic receptor? Just one word I need. Why? Tell me why it is a muscarinic receptor. It is binding to acid. We are studying the cholinergic receptors. Okay. Now, these cholinergic receptors, they are called as cholinergic because they bind to the acetylcholine. Then why these names nicotinic receptors and muscarinic, from where they have derived, from where these terms have been derived? Tell me. Yesterday we have discussed this. Just one word and the answer is done. What is muscarinic? Muscarin? All of you can hear me, right?
This
Lecture.
conference
So,
will now be recorded.
okay. So, I was continuously asking the question, but I was not getting any reply. So I thought, what is happening? So, okay, the question which I was asking was that why it is called as a muscarinic receptor. What is this muscarin? This is muscarin. So why it is called as a muscarinic receptor? Even though it is a cholinergic receptor binding to acetylcholine, why it is called as a muscarin receptor? In just one word, can you give me the answer? What is this muscarin acting as? Tell me. Right, right. It is acting as a agonist. Okay, it is very good, Vidushi. It is acting as a agonist. See, we have already discussed this yesterday that ligand is a natural one. What is the ligand for this cholinergic receptor? It is acetylcholine. But the scientists, when they were working on these receptors, they have used different ligand, and we should not call this as ligand because ligands are the ones which are present in our body. So they have used some structural similar forms like nicotine and muscarine. Okay, and the term for this is what? Agonist. Okay, why they are called as agonist? Because they can activate the receptor. Which receptor? Cholinergic receptor. Even if acetylcholine is not present, we can take this agonist and we can do our experiments, and we can study that what is actually happening inside the cell with the help of these agonists, and what are antagonists which will block these receptors. Simple. Understood, everyone? And here you can see that it is spanning, it is membrane spanning domain. What is the key thing which you have to mark here is that it is bind to a protein intracellularly in the cell, inside the cell. See, this is extracellular, this is intracellular. Inside the cell, it is bind with a protein which is called as a G protein. Can you see the location? This is very, very important because for 2 marks question, it is asked that where is this G protein located, in the transmembrane, in the extracellular matrix, in the cytoplasm, or in the intracellular transmembrane domain. See, this is linked with the transmembrane. It is linking. It is linked with the transmembrane, but it is present in the intracellular space. Very, very important. It is present in the intracellular space. Okay, you have to mark this thing, and this is called as G protein. Why it is called, what is the function, we will see. But in this slide, you just note the location and what is this G protein. Something which is called as G protein is attached with this receptor, the 7 membrane spanning domain receptor, and this here they are called as the alpha helix. Okay, these are the alpha helix. Forget all these things. We don't want these things. We will study all these things in the subsequent slides, and also when we are doing the cell signaling portion. Okay, there it will be dealt in great details. So just here you have to mark that it is linked with this G protein. G protein के साथ ये bind है. Okay, this receptor is binded with the G protein. Fine. Now what is, now you have to listen very carefully, because from this slide we will study the functional mechanism of these G protein. First thing, wherever this GPCR is present, okay, GPCR, which is called as the G protein coupled receptor. This GPCR, they are important for 2 pathways. Okay, for 2 pathways they are important. The first pathway is CAMP pathway. We have studied what is CAMP, but I will tell you here also, when we were doing the lactose metabolism, at that place we have done CAMP. But you just remember here that there are 2 pathways to which this GPCR is functional. The first pathway is called as CAMP pathway, and the second pathway is called as phosphatidylinositol pathway. The terms may be a little bit difficult for now, but when you are revising, it will be very easy. And these pathways are like key things, because without these pathways, nothing can happen. So these are the basic pathway for everything. If you are talking for growth, proliferation, these are the main pathways. Okay, so your G protein coupled receptor is mediating 2 main pathways. The first is called as the CAMP pathway, and the second is called as the phosphatidylinositol pathway. If you see this receptor has this binding domain where the acetylcholine will come and bind, because it is a cholinergic receptor. And as soon as this acetylcholine is coming and binding, sorry, there will be a structural change in this receptor. Okay, there will be a structural change. Okay, there will be some conformational change in this receptor. Now, what is linked with this receptor just intracellularly? It is called as a G protein. Okay, it is called as the G protein, which is linked with this receptor. As soon as there is little amount of structural change or conformational change in this receptor, this G protein will have a drastic change. Okay, now what is that change? If you see this G protein, it is made up of 3 subunits. The first is called as alpha subunit, beta subunit, and gamma subunit. So the G protein is made up of these 3 subunits. Fine, alpha, beta, and gamma. As soon as there is a structural change in this G protein, what it will do, it will break. Okay, and beta and gamma will be attached with this membrane. Okay, will be attached to this receptor. Sorry, not with the membrane. It will be attached to this receptor, and this alpha will be dislocated from this. Okay, it will be dissociated from this. And this alpha subunit is very, very important because it mediates all the internal function. whatever the pathway's next target is, it will go and show its effect on that target. So beta and gamma are not that important for the GPCR to function. It is alpha subunit. Beta and gamma are present to hold it and to give it a, it has to be in the natural form, something to hold it. So it is like it is holding this alpha subunit, and as soon as it is like something clip. Okay, beta and gamma are clips which are holding this alpha subunit. As soon as there is some structural change, this clip will open and it will release this alpha subunit. Now this alpha subunit is very, very important because the one very important role it is playing is on the potassium channels. Okay, it is playing a role in the potassium channel. But whether this acetylcholine is directly linked with the opening, tell me yes or no. This acetylcholine is directly linked or indirectly linked? What do you think? Very good. It is indirectly linked. What is meant by direct? Direct means just the ion channel will be present here only. Okay, here only the ion channels will be present, and as soon as this acetylcholine is binding, this channel will open and it will allow the passage of these ions. But this is not happening with this muscarinic. Okay, it is happening with the help of this G protein, and that is why it is called as a metabotrophic receptor or a GPCR or a G protein coupled receptor, because with the help of this mediator, the functions are happening. The first function is the opening of this channel. Okay, the second function is it is a very important for the activation of the enzymes. Very, very important. And the two very important enzymes are adenylyl cyclase and guanylyl cyclase. See here, if adenylyl cyclase it activates, it will convert ATP to CAMP. Okay, ATP to CAMP, and this is your first pathway which this GPCR is activating. I just told you it activates the CAMP pathway. So how it will activating this CAMP pathway? It will be activating because this alpha subunit will go and activate this adenylyl cyclase, which is present on the membrane only. Very, very important. See, the location of the enzymes is very important because it is asked in your 4 marks question. Targeted. Okay, 4 marks question, it is asked that what is the location from where this enzyme will move, from here to here, from where it will move. Okay, so these things you should remember. This is also a membrane located enzyme, this adenylyl cyclase. And this adenylyl cyclase, what is the substrate for this adenylyl? From the name only you can suggest it is ATP. And what it will give, this adenylyl cyclase will break this ATP into cyclic AMP or cyclic GAMP. Fine. So this is its second function. Second function, and a very important function. All the functions are very, very important. Okay, but these are the direct functions of these alpha protein. Direct functions. Now these functions, they can be direct or they can be indirect. They can activate more intracellular enzymes, like they can activate the kinases. Now, from the name, can you tell me what is the role of kinase? Kinase. What does kinase means? What is the role of kinase? Kinase means it will do very good. Phosphorylation, right? It will, there will be substrates, and on that substrate, it will go and phosphorylate. Now, as soon as that phosphorylation happens, either the product will be active and it will go to the next target and it will phosphorylate it, or it will go inside the nucleus and it will act as a gene transcription. If suppose it is a transcription factor and the transcription factor is activated by phosphorylation, that transcription factor will go to the nucleus and it will activate the gene transcription. or it may be inhibitory phosphorylation also. As the function present in the cell, they will mediate their whatever is their role on that target proteins. Fine. So these are the important things of the GPCR, that what are its role. We have to see these two. These two are important and these two are indirect. Okay, these two are direct, something which we have to study. Understood, everyone? What is this? What is this G protein? It is made up of 3 subunits. This is asked for 2 marks question. Okay, what are the subunits? One more thing is that, okay, in the next slide I will tell you that. So it is linked with this protein. As soon as the acetylcholine will come and bind, there will be structural change. The alpha subunit will be dissociated. The alpha subunit will help in the opening of these channels or the activating of these enzymes, activation of the kinases and the gene transcription. Understood? Okay. Now, let's see that what is the CAMP pathway. Okay, we have to know this pathway to understand what is the role of this muscarinic on this pathway. We have to understand that what is this pathway. This is your GPCR. Okay, this is your GPCR protein, a 7-spanning membrane domain protein. Here, what is present? It is linked with the G protein. It is linked with your G protein. Now, this G protein, it is made up of alpha, beta, gamma. Now, this adrenaline, norepinephrine, acetylcholine, whatever is the case, okay, here the example of adrenaline is taken, but you should not be confused, okay, because this GPCR, they are mediating many functions. They are activated by many channels. If specifically we are talking of muscarinic, then it is for acetylcholine. But if we are talking of GPCR, GPCR is a very big family. Okay, so just don't confuse with this. Just take it as an agonist. Okay, it is an agonist. And as the agonist is binding with this, what will happen? There will be change in this and your alpha subunit will be dissociated. Now, why is only alpha subunit is being dissociated? Why not beta and gamma? Because alpha has something very special in it, which is called as a GTPase activity. Okay, a GTPase activity is present with the alpha subunit. Either GDP or GTP can bind. Okay, either GDP or GTP can bind. If it is inactive, just when it is binded with the receptor, the alpha subunit is bind to GDP. GDP is inactivating the alpha subunit. But as soon as the agonist is binding, so what will happen? There is an inherent Jeff property activated. What is this Jeff property? That is exchange factor, nucleotide exchange factor, guanosine nucleotide exchange factor. This GDP will be converted to GTP, or in other words, the GDP will remove and the GTP will come and bind with this alpha subunit. As soon as this GTP is coming and binding to this alpha subunit will be like activated, and it will go and do its whatever is its function. Whatever we have seen, it's so many functions. So for that to happen, it is important that alpha subunit should be bind to this GTP. Now tell me one thing. If suppose this alpha subunit is mutated, okay, mutated, and a constitutive mutation is present, what will happen to this alpha subunit? What will happen? Suppose it is, I'll reframe the question. This alpha subunit is binded with this GPCR. Okay, this 7 membrane spanning domain, that is GPCR, it is bind. And in this G protein only, there is mutation in this alpha subunit. What will happen? Tell me. Very good, Vidushi. Right. It will not be able to bind with GTP. Okay, it will not be able to bind with GTP. Or there can be another case also. Suppose if there was GTP and then the mutation has happened. Okay, so there the case will be, it will be continuously binding with GDP and it will not dissociate it, and it will bind with GDP. In that case, there are two cases. Either it will be permanent inactive or either it will be permanent active. Both these cases leads to cancer. Okay, this G alpha proteins, they are very, very important because they are important in the metabolism. When you see this, they are indirectly, they are linking with this gene transcription, okay, gene expression. And wherever this gene expression term is coming, that means it can lead to cancer. Okay, so this is a type of analytical question which is asked, and it has already been asked in your CSIR exam. So you should keep a note of this, that what will happen if the mutations are occurring on this. Fine. So suppose this now in normal situation, this alpha is bind to GTP. Okay, now what is its function? It will go and on the membrane, adenylyl cyclase is present. It is a membrane enzyme. It will go and bind with this adenylyl cyclase. And as soon as this adenylyl cyclase is bind with this G protein, that is alpha subunit, not G protein. Now it is called as alpha subunit. What it will do? It will convert ATP to CAMP. Okay, it will convert ATP to cyclic AMP. What is the function of cyclic AMP? Cyclic AMP will go and target protein kinase A. Okay, it will target protein kinase A, which is a kinase protein. Okay, now these kinase protein will phosphorylase its substrate, which may be glucose or glycogen, whatever may be. Another enzyme, it may go and target, and then it will directly, it will be involved in the metabolism. So you just have to remember till here that GPCR, agonist is binding, change in the GPCR, alpha subunit is removed, GDP is dissociated, GTP is bind, it will go and bind with this adenylyl cyclase, ATP will be converted to cyclic AMP, and cyclic AMP will phosphorylate protein kinase A. This is a activation. Okay, why activation? Because the first step is leading to the first step is leading to the whatever the signaling proteins are present in the down membranes, they are being activated. So this is your first pathway, which is called as the CAMP pathway. This is called as the CAMP pathway. Now, did you understand why it is called as CMP? Because here this CMP, which is called as a second messenger. Okay, why? Because the first messenger is this. This has bring the first message. And this first message has led to the opening of this second messenger, which is CAMP. And this second messenger will target the kinases. And then those kinases will go and show its effect on the subsequent kinases or its effector, whatever the substrates are present. But you have to remember that cyclic AMP, A for A, okay, because there are so many protein kinases. Protein kinase C is also present. But this is again a second 2 marks question, that cyclic AMP phosphorylate which protein kinase? It is protein kinase A. And how you will remember? A for A. Cyclic AMP will phosphorylate protein kinase A. Understood? This was your first pathway. Any doubt in this pathway? Write down the flow chart. Okay, small in short, you can make the diagram and write down the pathway. Very, very important. This pathway is very, very important. Now let's see the second pathway. Or okay, one more thing. Just now I told you that this is an activating. The previous slide which we were talking about, this is an activating receptor. Now this alpha subunit, actually they are of different types. It can be stimulatory. Okay, the steps, they are written in forward slides. Okay, they are written, they are present in this slide only, in this presentation only. But usually what I do now, I don't read from there. With the diagram, it is easy to make you understand. And for me also, it is easy to explain. Now, that's why I'm doing. Okay. So this is an example of a stimulation. Okay, for example of stimulation, if this alpha subunit is of stimulatory type, okay, if it is an of stimulatory type, it will give this type of response. But there are certain alpha subunits which are inhibitory type. Okay, inhibitory type. So what will be the response for that? Will these things will happen if the alpha subunit is inhibitory type, and even though it is bounded with the GTP, will these things will happen? This alpha subunit is specific for stimulatory type. You have to understand this thing, that same receptor, same subunit can give both the responses, inhibitory, stimulatory, and on what basis that is decided? On the basis of this, on the basis of the agonist, on the basis of which type of agonist is binded with this GPCR. Because we have studied that acetylcholine can give excitatory response, but there are certain neurotransmitters which gives inhibitory responses also. So in that case, if the same pathway is followed, if the same GPCR pathway is followed, then how it will happen? Because we are studying here that alpha subunit is binding with the GTP, and if this is binding with the GTP, definitely it will go ATP to cyclic AMP. Fine, but there are differences in this alpha subunit. Alpha subunit can be of stimulatory type and it can be of inhibitory type also. Can you see the types here? See, stimulatory alpha, inhibitory alpha. Okay, so this stimulatory alpha will give a stimulatory effect, and this inhibitory alpha will give you a inhibitory effect. Can you see this? GSGTP and GIGTP both are linked with GTP, but then also they will give a different effect. So from here, one thing you should be very clear: always phosphorylation will never lead to activation. Many phosphorylations are showing inhibitory effect also, and the same protein can have diverge functions. Okay, they can have diverge functions, and on what basis this stimulatory and inhibitory G proteins are categorized? There will be different catalytic centers. Okay, a different catalytic center will be present, and that is why they are acting as a different molecule. Even though they are same, the name is same, but they are acting differently. Understood this? Everyone, Priyama, are you getting the things? She's present na today, Priyama? Fine. So this was your first pathway. Okay, CAMP. This was your first pathway. Let's see what is the second pathway. I told you it is called as phosphatidylinositol pathway. Why it is called as phosphatidylinositol pathway? Because it acts on this phosphatidylinositol. That is why it is called as a phosphatidylinositol pathway. What will happen as soon as the agonist is binding with the receptor? What is the receptor? GPCR receptor. Okay, muscarinic receptor, GPCR receptor. Okay, this GPCR, they can have two different mode of pathway. The first was cyclic AMP pathway. The second is this phosphatidylinositol pathway. So in short, this phosphatidylinositol is called as PI. This PI is broken and it gives you diphosphate. Okay, it gives you a diphosphate structure. And this diphosphate structure, you should remember the name here. Okay, this is 4,5-diphosphate or biphosphate, whatever it is. This diphosphate, what is its the subsequent end products? The first is called as diacylglycerol and the second is called as inositol triphosphate. All the names are given in the subsequent slides. Just here, listen what I'm telling. It is called as DAG and IP3 molecule. Okay, now see, one thing you should note here, it is linked with the membrane. It is linked with the membrane. So there is a protein, okay, phosphatidol inositol, sorry, a substrate phosphatidol inositol, which is broken down, and then it gives you two products, which is called as DAG and IP3. Now, in your CMP pathway, what was your second messenger? CAMP. Here in this pathway, the second messenger are these two, DAG and IP3. Okay, these are two second messenger, because these second messenger, now in very literal terms, if I'm telling you what is the role of the messenger, they will go and give the messages. Fine. So that is why they are called as second messenger, because your first messenger has already done his task. Okay, it has to give the signal to this phosphatidol inositol, and this phosphatidol inositol has given its end product, the DAG and IP3. If this DAG and IP3 are standing just like that, the message cannot go forward. Okay, that is why this DAG and IP3, they are called as second messengers, because now see what function they will play. The first, let's take this DAG. Okay, DAG is always functioning on the membrane. Very, very important. It is functioning on the membrane. Okay, and with what it will do, it will go and target this protein kinase. This PLC is phospholipase. Okay, whatever is shown here now, it has been shown in the diagram here. Okay, this PIP2 is this PIP2. Okay, this PIP2 is broken by the phospholipase, and this phospholipase is broken in DAG and IP3. Both are second messenger, but the role of these second messengers is different. You should know the function of the each second messenger. What was the function of the cyclic AMP? It will convert ATP into cyclic AMP. Simple, adenylyl cyclase. Actually, it is functioning on the adenylyl cyclase. What is the role here, DAG and IP3? First is DAG. DAG will go and target protein kinase C. Just now I was telling you that cyclic AMP target is protein kinase A. Okay, and DAG target is protein kinase C. This is important. You should write down in your notebook that which kinase is activated by which enzyme. This is asked in your exam for 2 marks. And the second one, so now it has given its message forward to this protein kinase C, and as the name suggests, protein kinase, because it is a kinase protein, it will phosphorylate its substrate, and majority of the phosphorylation will lead to the nuclear gene transcription. So in this way, the message is going forward, and it is reaching it's reaching the gene which is present in the nucleus. So that is why it is called as a second messenger. So first role is finished. That is for diacyl glycerol. That is DAG. Okay, DAG. DAG will function on protein kinase C. Simple. DAG will function on protein kinase C. Now, what is this IP3? Now, how you will remember this? Now, you should remember this, that the number, because here the number is present, this number will activate a number. Okay, this IP3 will activate a number. And what is that number? This IP3 will go and they will bind with this calcium channel. See this number. Okay, so this IP3 will go and bind with the calcium channels, and that also the calcium channels which are present on the endoplasmic reticulum. Very, very, very important, because this question is asked so many times. Okay, this IP3 will go and bind with the calcium channels which are present on this endoplasmic reticulum, and inside the endoplasmic reticulum, calcium ions are present. And as soon as this IP3 go and bind with this calcium channel opens, and the calcium from the endoplasmic reticulum, it comes into the cytoplasm. Each and every sentence what I have mentioned is important, because they will ask you what is the role of IP3. To which organelle it will bind. Calcium channels are present on which cell organelle? The calcium channels, if IP3 is binding to the calcium channel, from where the calcium channels will come? Okay, what is the direction of this calcium? So what is the direction of the calcium? The calcium will move from endoplasmic reticulum to cytoplasm. It will not move from cytoplasm to endoplasmic reticulum or from cytoplasm to extracellular or from one cell to another cell. No, from endoplasmic reticulum to cytoplasm. Okay, and these calcium, then they will help in the transcription of the genes. So that is why these are called, we have to study this pathway. That doesn't matter that what is the higher or the lower concentration. You have not attended the previous lectures, na Asha? Now see, these are depending on the cell's requirement. Okay, here actually if I see here, the thing is that inside the endoplasmic reticulum, the calcium is present at a higher concentration. Okay, and from this higher concentration, the calcium is coming out into the cytoplasm. But what is the case now? Sometimes there are calcium channels which are present here on the membrane also. And because of this agonist binding, these calcium channels will also open. And from the extracellular space, the calcium will come inside the cytoplasm. So even though the cytoplasm calcium ions are present, but since this pathway is activated and this IP3 has been released, the role of IP3 is only one thing. They will go and bind with this calcium channel. And once this calcium channel has been opened, the calcium has to be released. Okay, the calcium will be released. So it doesn't matter that the calcium are present here or not. Just focus on the pathway. Now, why all these things are so intricate? Now, that is why cancer is a multigenic and a multifactorial process. Okay, so that is why when you are studying cancer and its genetics and everything, you have to look at each and every thing. And that is why the treatment is also very difficult, because if you block this, the cancer cells are so intelligent, they will open the other calcium pathway. So if we think that, okay, now this calcium channel has been blocked and the calcium release has been stopped, so the nucleus gene transcription will not happen and proliferation will not happen, because mostly in cancer, the main problem is the proliferation. The cells are continuously dividing. But these cancer cells they are very, very, very smart and they are very cunning. So what they will do, they will open some channels here. So the calcium will start coming. And whatever is the target for this calcium, they don't recognize, they don't distinct that this calcium has come from endoplasmic reticulum or from the membrane. They will not differentiate on the basis of that. Understood? So, did you get the functions of these both second messenger? They are also called as second messenger. It is not like that only cyclic AMP is called a second messenger. DAG and IP3 are also second messenger. So 1-2 marks question which can be asked is that from the following list, which is not a second messenger? And they will give you DAG, IP3, PIP3, cyclic AMP. So from that you have to mark which is not a second messenger. Understood? Understood these two pathways? Whatever I was telling till now, it has been written here. Okay, all these things are the same thing which I was discussing till now. One thing I've missed out was that now once alpha subunit has done its function, okay, so what will happen? It has to go back to its place. So at that time, what will happen? There is a GTPase activity which is inherently present in this G alpha subunit. Okay, as soon as its function is done, this GTP will again be dissociated and GDP will come and bind. This GTPase will dissociate this GTP and the GDP will come there. And when the alpha subunit is bind with this GDP, this beta and gamma subunit, they will go and hold this alpha subunit and they will take it back to the membrane receptor. But the location is intracellular. So whatever is the function happening after the dissociation of this G alpha subunit, that is very critical. Till how long this G alpha subunit is free. Okay, only till that time the effector proteins or the next target proteins will be functional. as soon as this G alpha subunit comes back to its place, to its location, all the process will stop. And how this G alpha protein will get back to its place? Because there is a GTPase activity present. And when this GTPase activity will function, if suppose the acetylcholine is dissociated, once the message is not received, this G alpha subunit don't know that what message to give forward. So it will come back to its place. Understood? So the time is very important. If you want the transcription to happen at a larger time or for a larger event, then this G alpha subunit has to be dissociated to a longer time. So for that, this acetylcholine, it has to hold the receptor for a longer time. So acetylcholine, I have mentioned in the last slides also, sorry, yesterday, that this acetylcholine, it is being degraded with the help of this acetylcholine esterases. Okay, suppose if this acetylcholine esterases is blocked, what will happen? Somehow there are some toxins, which we will see in the slides, that there are some toxins present, or some venom or some poisons which are present. What they will do, they will go and they will break down this acetylcholine esterases. Once this acetylcholine esterases is absent, acetylcholine will not be degraded, and acetylcholine will bind the receptor very strongly. Once the acetylcholine is present, what will happen? The inside train, it is running and running and running. Okay, understood, everyone? So what is actually the response of this muscarinic receptor? Whatever kahani we were studying till now, we have to apply here now. Okay, so what are the responses of this muscarinic receptor? If suppose acetylcholine is coming and binding to this muscarinic receptor, what will be the response? The first response is inhibition of adenylyl cyclase. Whatever we have studied, activation, G alpha, GS, GI. So what is the role of this muscarinic? Whether, if suppose acetylcholine is coming and binding, what will be the inside action? What will be happening? Tell me if you have understood this. Inhibition of adenylyl cyclase. Just from this word, what you understood? Whether the train will run? Yes. Yes. Right. Right. Not that, right, Vidushi. Okay, it is inhibitory. Alpha subunit won't bind to GTP. No, Deepshikha. No, 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 no. Just now we have studied the G stimulatory and G inhibitory effect. In both the cases, it was binded with the GTP. Okay, in both the cases, it was binded with the GTP. But the role here is, if suppose it is stimulatory and it is binding with the GTP, it will go and activate adenylyl cyclase, and this protein kinase will be, this cyclic AMP will be formed. But inhibition of adenylyl cyclase means it will definitely go and bind with GTP. But the subunit is different. It is not a stimulatory subunit. It is an inhibitory subunit. And when that inhibitory subunit is bind with GTP, it will never go and activate that adenylyl cyclase. And once adenylyl cyclase is not activated, whatever is the kahani going behind that will be stopped. So if acetylcholine is binding with this muscarinic receptor, it is showing an inhibitory effect on the adenylyl cyclase pathway, that is CAMP pathway. Very simple. In one word, if I want to tell you in one line, CMP pathway will be blocked. If acetylcholine is binded with this muscarinic receptor, it is showing an inhibitory effect. Got my point, everyone? Or you are confused? Right? So if one pathway, now GPCR can activate two pathways, CMP pathway and IP3 pathway. Okay. Now, if CMP pathway is blocked, definitely the other pathway will be stimulated. So your muscarinic receptor, they are blocking this adenylyl cyclase pathway, but they are stimulating this phospholipase C pathway. And we have seen that what is the pathway, what are the mediators, and how the pathway goes. So same thing is written here. Okay, same thing is written here. Just you have to know that CMP pathway is blocked and PI3 pathway is stimulated. Fine. The same thing, what we have studied till now, everything is written here. Okay. Asha, it is called as muscarinic receptor because the agonist called as muscarine can activate this pathway. Okay. See, it is a cholinergic pathway. Okay, it is a cholinergic pathway. But in our body, the ligand is acetylcholine. But the scientists, when they were doing, they have done it with different chemicals. So they found that this chemical called as muscarine, it can go and bind with this receptor and it can activate the IP3 pathway. Now, acetylcholine is not present, but even though the pathway is activated now, so that is why it is called as muscarine receptor, to distinguish it from the nicotine receptor. Okay, because there is something called as nicotine receptor and that is activated by nicotine. Fine, but both are cholinergic receptor because in our body both are activated, both can bind to the acetylcholine. Both can bind to the acetylcholine. Understood? Okay. And what is the third important function of this muscarine receptor? It will activate this K plus channel. It will activate this K plus channels. Okay. So once the K plus channels are open, all the K plus which are present inside the cell will start moving outside. All the K plus which is present inside the cell will start moving outside. Now tell me, what is that term called? What will happen? If all the K plus is moving out, what will happen? And nothing is going inside. Na plus is not going inside. Only K plus is going outside. What will happen? Very good, Vidushi. Very good. Hyperpolarization will happen because आपका जो membrane potential है, what is the membrane potential of your cell? Minus 70 millivolts. Okay, and how that minus 70 millivolt is being maintained? Because of this potassium, there are leaky potassium channels which are present, and because of that, there is minus 70 volt charge which is present on the cell. But if all these K plus channels are moving out, this minus 70 will go into more negative sign. More negative sign. And what is the more negative sign? It is hyperpolarization. And if it is having positive sign, it is called as depolarization. I have told you that how you will remember this hyperpolarization. Negative people are always hyper. Okay, negative people will always inhibit the things. Negative people are always blocking everything. Okay, so if the cell is going negative, it is hyperpolarizing, and the effect will be inhibitory. The effect will be inhibitory interactions. Okay, it is not excitatory. It is an inhibitory interaction. Okay, this interaction is an inhibitory interaction. Understood why? Because K plus is moving out. And how this is happening? Because your GPCR has bind to this acetylcholine. And the reaction which is happening inside the cell is what? K plus channels are open and all the K plus is moving out. So your cells are going in an inhibitory reaction. That means you may have lower heartbeats. You may have this smoothening, what is called as relaxing of the muscles. Okay, so there is a chemical or a toxin which is called as atropa belladonna. Very important. The name of this chemical is very important. And what is this atropa belladonna going to do is that it binds with this muscarinic receptor. Okay, this atropa belladonna binds with this muscarinic receptor. Now acetylcholine cannot bind. And this atropa belladonna, it is an antagonist. So opposite function it will do. It will do opposite function. And what will happen? It will inhibit this function and there is pupil dilation. For eyes now, when you are going for the dilation of these pupil, okay, so at that time this atropa belladonna is given. Okay, this is an antagonist, atropine. This is very important. This is asked in your CSIR, and there is a possibility that it can be asked again. So remember the name of this chemical and to which it binds. It binds to the muscarinic receptor, and what is its role? Pupil dilation. Your eye has the central portion called as pupil. So to get light more, this dilation of pupil is done. But because here the muscles are holding your pupil very tightly, so it will not open, right? But this atropine, what it will do, it will relax all your muscle and your pupil will open up. Understood? Got the point? Fine. And already we have mentioned this acetylcholine esterase. You just read this. We have discussed all these points. Now there here also some chemicals name are given. See, this is one of the inhibitor, and there are some insecticide. So what is the role of this inhibitor? This isopropyl fluoroesters, you just don't remember this name, okay? Don't remember this name, but that atropa name was important. But you should know that what is actually this isopropyl is doing. It is actually is inhibiting the function of acetylcholine esterase. What is the function of acetylcholine esterase? It will break this acetylcholine. Okay, it will break this acetylcholine. And suppose if this enzyme is not present, acetylcholine will always be active. Okay, this will always be active. So the 4 marks questions are targeted from here. Okay, the 4 marks questions are targeted from here. You don't remember the name of these this thing. No need. But what will be its effect? You should know the effect, because as a research person, you should have an analysis. Okay, if this drug is an acetylcholine esterase inhibitor, what will happen? So those happenings will be given in your option, and you have to mark the correct option. Fine. Also, there are some important ones which you have to know. Now, this thing we all have done. You don't have to need this. Okay. See, this is one of the table which I have made. You should know the names and what is the action. No need of reading this clinical uses. That is not important. But definitely the drug and what is its action. It is very, very important. Very, very important, because it is asked in 4 marks for match the column. Okay, can you see here? Atropine, scolopine, amantane, they are all blocking this muscarinic receptor. Blocking the muscarinic receptor. This etheric, taccharin, they are blocking the acetylcholine. They are blocking this acetylcholine breakdown. That means they are working on this acetylcholine esterases. And this bethanocol, it is mimicking as acetylcholine. So if suppose I am introducing this drug, what it will do, if suppose acetylcholine is not present, acetylcholine is not being synthesized, because no action potential has been sent. So if action potential is being sent, only in that case this acetylcholine will be synthesized. But if I am administering this drug, I don't need this acetylcholine. This drug will go and bind with this acetylcholine receptors, and it will start its function. Okay, now see, what is its function? Used to treat the urinary retention. In some of the cases, some diabetic persons, or in certain conditions, there is problem in holding of this urine. Okay, so why there is a problem? Because the acetylcholine is not released. Okay, because acetylcholine has parasympathetic role. It will have an inhibitory effect. So in that case, these drugs are being administered, because they will mimic this acetylcholine and they will give the same function what this acetylcholine is giving. So you should know the name of the drugs and the action. Okay. This is also very important. And from here, the questions already have been asked, and there is a possibility that the questions can be asked. Neurotoxins, this slide is very, very important. You should know that which toxin is working on which channel. Can you see here? Almost all the channels are Na plus. Okay, all the channels are Na plus. There is only one toxin, dendrodotoxin, which is acting on K plus. So this thing is very simple. How you will remember this chart? This chart is very, very important. You cannot miss this chart. Even though if you are not reading this thing, you cannot miss this. Okay. And how you have to remember this? First thing is that all are working on the Na plus. Now, whether they are opening or blocking, that is another story. But all these toxins are working on Na plus. So if you are not remembering the name of these things, now till this time it is okay, but you know that okay, dendrotoxin is one starting with a D, which is acting on the K plus channel, and what it is doing, it is opening the K plus channel. So dendrotoxin with the K plus channel. If I see all these things, let's see that which are opening the channel. Okay, which are the things which are opening the channel? Can you see? There are only two, betradine and batrachotoxin, which are opening the Na plus channel. So this is BV opening the channel, BV. Okay, BV are opening the channel. And the rest two, they are blocking the channel. Simple, very simple. I hope you have understood what I meant to, what I wanted to give you here. Is it easy now to remember? Okay. And what is asked in your exam is only these things. They will give you at one side neurotoxin and the effect on the other side, and they will ask you either in the match the column, or they will give you the name of this toxin and they will give you all for this: permanently open, permanently closed, open, or K plus channel. Like this they will give you, and from the options you have to select. So whenever you are getting these kinds of tables, now you should never miss that table, because these tables are very, very important. Very, very important, because from these tables, 4 marks and 2 marks, both the questions are targeted. And these tables are easy when you are using the mnemonics. But if you see the syllabus, it is wide. Okay, it is broad. So many mnemonics will be present at the end of your syllabus. So it is important that you revise. Because if I'm telling BV, okay, BV is easy, you can remember that. But sometimes some mnemonics you need to revise and revise. Only in that case you will remember that. Fine. So let's do some of the questions. 10 minutes, okay? Only 10 minutes. First is, depends. Okay, Asha, what is the quantity what you have administered? Because all the time pupil dilation is also not good, na? It is, if it is used as for a drug basis, the quantity what is being administered is important. Okay, everything is poison if it is taken in a higher amount, right? And everything is medicine if it is taken in a controlled amount. So that is the thing. So it depends on the basis of the question which is asked. Fine. So electrical impulses, it is for all the questions are for the ones who are attending the lecture from the first day. Now tell me. The others, if you know the answer, definitely give me. Okay, fast. Electrical impulses gather and accumulate in which part of the neuron? In which part of the neuron do you think the action potential will accumulate and then it will move forward? Dendrites, axon hillock, axon terminal branches, nodes of Ranvier. For your ease, I will make a diagram, bad diagram, but okay, you can get an idea. This is your neuron. Apparent neuron. Okay. So these are your dendrites. This is your cell body. This here the axon hillock is present. These are the axon terminal branches, and your nodes of Ranvier are present here. The nodes, these are your nodes of Ranvier. So now tell me, by looking at this figure, what do you think where these electrical impulses should accumulate? Tell me fast. Okay. Okay. See, nodes of Ranvier, nodes of Ranvier, when it will function, when your action potential has reached till here. Only after that, there will be a saltatory conduction. Okay. But what is the question here? Initiate, where it has to gather. Okay. Nodes of Ranvier is for faster electrical impulses, because here nodes of Ranvier are present. These are gaps. So your action potential will jump like anything, and they will go very fast. But where it has to accumulate? It has to accumulate here. Here it has to accumulate. It will take the signals, the dendrites will take the signals, and it will accumulate here, and after that the signal will pass. So it is axon hillock. Vidushi was right. It is axon hillock. You got my point, everyone? New ones also. Fine. Okay. Second, very easy. This is very easy question. What is largely responsible for the negative resting membrane potential? What is responsible? I think without looking at the option, you should tell me. Right. It is potassium leak current, because whenever membrane potential is asked, yes, only one ion is important, that is K plus. K plus is important, which is maintaining your resting membrane potential. Right, it is potassium leak currents. Which of the following nerve fibers are typically all unmyelinated? Okay, I may have missed this point, but we will discuss here in this question. The answer is C fibers. Okay, C fibers. C fibers are the nerve fibers which are unmyelinated. They will never have this myelin sheath. Myelin sheaths are present on this alpha, A fibers. They are highly myelinated. Okay, they are highly myelinated. A fibers are highly myelinated and C fibers are unmyelinated. Fine. So note down in this thing in your notebook. Next question. Okay. A alpha fibers typically conduct action potential to signal which of the following? Tell me. If you don't know the answer also, by looking at the options, you can tell me the answer. If you don't know the answer also, but you know this thing, that these fibers help in the conduction of certain messages. Okay, so some messages it will help. So there will be some action which will happen. If some action, suppose if I give you one example, I was walking on the road and one thorn has pricked in my legs. So what will be my immediate effect? Pulling off my leg. So which fiber will take that? Because these are alpha fibers, and just now I told you that they are myelinated fibers. So they will take the message. Yes, Mansa got the point. It is skeletal muscle. Now see here, if suppose, how I will analyze the answer now. Suppose it is CNS that the body is in contact with painful stimuli. But your second option is also giving CNS. Two options cannot be right now. You have to mark only one option, that is which is right. If CNS, if it is acting on this CNS, it will be acting on this CNS also. How you will mark two options? So definitely A and B are wrong. Now, if you see this third option, viscera to increase gut motility. Motility, do you think fibers work on motility? No. It is only this skeletal muscles to contract. Understood how you will negate the options? It is D. It is D. Understood, everyone? Okay. Local anesthetic drug, very, very important, can be asked in your 4 marks question. Okay, I have written here only in 1 line, but there it will be given in 4-5 lines, making it confusing. But the answer will be in only this options. Okay, local anesthetic drugs work with which mechanism? You know, na, anesthesia, what they do, anesthesia will okay, see, anesthesia will help you in diminish the pain sensations. So see these things and tell me. Definitely this is a confusing question, but if we analyze, it is easy. Only one answer I have got, C, B. Okay, if I was also there, Priyama, I would have marked B only, neurotransmitter, C. Okay. Okay. No one is giving D answer. So, okay, fine. That means you have understood. Okay, Vidyushi, tell me why A. You are right, Vidushi. It is A. Okay. Vidushi is right. It is A. It was a guess or because everyone was telling B, C, so you marked A. Now think. Okay. This drug, what is its role? It is not moving the pain sensation. It is not moving the pain sensation. Suppose if this neurotransmitter blocking the neurotransmitter release, fine. If I'm telling you that this neurotransmitter release is blocked, the name of the neurotransmitter is not given. Okay. Here they should specify the name because there are so many neurotransmitters which are present. Fine. Here also, if suppose blocking the nicotinic acetylcholine receptors at the synapse, if they are giving, what will be its ultimate effect? If suppose I am blocking this nicotinic acetylcholine receptor, what will be its ultimate effect? What is taking your action potential forward? The forward taking is this. The channels are opening. The channels are opening and then the action potential is going to your brain and the brain is giving you the message, okay, you are feeling the pain. Fine. Your anesthesia, what they will do, they will block this sodium channels so that the action potential is not reached to your brain. This option though, it is wrong only. So remember this, your anesthetic drugs are working on sodium channel. Now, whatever is the anesthetic drug, A to Z, whatever is given in your question, you should know only one thing, it is working on Na+ channel. Got the point? What is the role of Schwann cell? Very, very easy. What is the role of Schwann cells? Okay. One answer I got A. Vidushi is also telling A. Rest, Priyama, Deepshikha. Deepshikha will not be able to answer because she was not there. See, right. This time Priyama is right. Enhance the speed of the action potential. Now see here, you have seen thermal insulation and you have marked. Insulation is right, but whether it is thermal, is there any thermal thermal thing going on in your brain? That is why you have to read the question very, very, very carefully. Yes, you are right that Schwann cells, they insulate the neuron, but electrical insulation. They do electrical insulation and not thermal insulation. They do electrical insulation. So what is the next best answer? The best answer is they speed up the action potential. It is an indirect answer. Okay, action potential actually it is through the nodes of Ranvier. But definitely what is the first thing is your insulation, but the insulation is not thermal, it is electrical insulation. Understood? So the answer here is C. C, enhance the speed of the action potential. Got my point? You have to be very careful while reading the question and the answer, because obviously first time when I was also solving, I've marked A, but then I saw that it is thermal. It is wrong. Thermal is not the word. Okay, last three questions. So are you getting familiarized that how the questions will be asked? I'm solving the question so that you know how to deal with the question. Definitely, it's not like that these questions will only be asked, but definitely you will get an idea that how you want to, how you will solve the question, how you will attempt an answer. That is very important, that how to attempt an answer. Okay, this is very, very easy. Tell me. The opening of the axon, I'm not able to read it. Axon membrane voltage gated potassium channels is responsible for which part? Very, very easy. Potassium channels open, what will happen? Single word, just you don't have to see also. Tell me. Prema, remember the action potential wala thing. Action potential, action potential, action potential, action potential. First, in action potential, what is happening? Depolarization, right? Yes, it is depolarization. But with which enzyme, with which ion it is happening depolarization? With which ion? First ion channel which is opening in the action potential. Which ion is opening? We have done it with the video also. Calcium? No, Mansa. You were not there, na? Right, Movnia, it is Na plus channel. Remember, this was the depolarization graph we were seeing, the action potential graph. Here the Na plus channels open, right, Anaga? Na plus channels open, and here depolarization is happening. So what is happening here? The action potential is coming down. What is opening here? Which channels? The K plus channel, the K plus channel. And this depolarization, what will be happening here? Repolarization. Remembering, we have discussed this thing. Now tell me the answer. I'm wrong or I'm wrong, right? Right. It is repolarization of the membrane. Just go back to your graph. Just go back to that graph and see. It was first Na plus D and then Re. I have told you A, B, C, D. Remember? So first word should be depolarization. And then D ka ulta Re, the repolarization. Here Na plus channel first opens and then K plus channels opens. A very easy question, and this, if you miss these types of question, so it will be difficult, because easy question will be solved by everyone. Okay, everyone. So you have to target easy question, and there will be categories now: easy, medium, and difficult. Okay, if you are able to solve all the three, you are in your top 10. But if you want to pass the exam, you should be able to solve easy and medium questions. Forget the difficult question, because there are so many options. You will have 35 in from 35 questions. You have to sorry, from 50 questions you have to solve only 35 questions. There are so many options you can choose, but the options of choosing 35 is very important. Fine. Which of the following is a drug that inhibits neurotransmission? Okay, you may not be knowing this, but when you read that graph now, that table, sorry, that table, you will get the answer. It is tuberculin. Okay, tuberculin. You will read, when you read that table now, you will get this answer. Okay, now tell me, this is very, very easy. Which of the neurotransmitter is doing inhibitory transmission? I have told you the example, and there is only one important neurotransmitter which is involved with this inhibitory one. Inhibitory. Tell me. Acetylcholine. Acetylcholine, though, it is doing excitatory also. We have seen just now. Excitatory also it is doing. It is involved with only inhibitory. Whether here it is mentioned. See, two words here you have to look: fast and inhibitory. If you are telling the acetylcholine, okay, acetylcholine, definitely it is going for inhibitory. You are right, it is inhibitory, but it is going through muscarinic channel, and muscarinic channel are slow channels. Okay, muscarinic channels are slow channels. Right, Mansa, it is GABA. It is GABA. Inhibitory, only one acid, only one neurotransmitter you have to remember, it is GABA. GABA is going for a fast inhibitory channel. It is opening chloride ion, Cl minus ion, and so it is giving a hyperpolarization. You are right. If suppose GABA was not written here, na, acetylcholine was right, but the word fast should not be there because it is going through a metabotropic receptor, sorry, through the muscarinic receptor. And muscarinic receptor, you know, it is bind with this G protein. So this G protein, the receptor, the first messenger will come and bind. Then this G protein will be dissociated. GTP activate will done. Then it will open the adenylyl cyclase. Then the functions will be done. So it is a slow. It is a slow. But if the word here is fast, it is only this. The answer is this. Without opening your eyes, you can mark GABA. Understood? And if suppose here the answer, the question is fast excitatory. Okay, then you have to think, because glutamate is also excitatory and these are also excitatory. But these types of questions are not asked. And suppose if these types of question asked, one thing you have to do is leave this question. You have to choose a next better question where you are not confused. Got the point? So we have done with today's lecture, and we have finished all the topics. If suppose something is remaining also, when we are revising, we will complete that. Fine. But from my side, we have done with this A part. Now see, the A part, if you see now words, the words are only 6 to 7. But did you see how the immense depth is needed if you are studying it for CSIR? Okay. You may be reading it for your M.sc and everything, but reading for a competitive exam is a different way. The same thing is read in your post graduation. You have to write the answers in your exam, but those answer writing will not help you in CSIR exam, these competitive exam. An average student, a below average student can also clear this competitive exam. Only tricks are needed. Okay, definitely certain concepts are needed, but tricks are the most important things. So I'm done with today. If you have any doubts from your side, you can ask me. I will share the slides today's also, yesterday also, but there is some difference. Yesterday also the same topic was there, dealt, but I have changed some of the slides, and these questions also I have added. So what you have to do now, you have to make a separate book for your question answers. Okay, write the question, because most PDFs also I am providing you. You can Xerox those PDFs. Just what I want is that now you make a separate file for questions. Okay, so suppose you have finished one topic, so you can go and solve the questions of that unit. If you solve it 2 times, now the questions will be like fixed in your mind. Next time, as soon as you see the question, you don't have to see the option also. You will just look for the right option. Okay, this is the answer. So, okay, this is option. Okay, this is the right answer. Fine. So make a separate place for your question answers. Don't mark it in your notes where you are writing the notes. Now, don't mix it there. Or you can do also. If you are continuously revising, then it is fine. Any doubts from your side? No doubts, na? Vidushi, you are doing very good, na? I'm happy with you. Just continue the way you are reading, and definitely you will be in our one topper. Everyone has that potentiality, but Vidushi is responding the most, and mostly she's doing it right. So everyone, I'm telling you, I know that for the first time it is a little bit difficult, but just you have to do one thing, revise. Okay, just think that, okay, for 2-3 months, I have to dedicate myself. After 2-3 months, if you are having a CSIR tag, these 2-3 months will vanish like everything. Just for 2-3 months. If you suppose family have, if you have family also, then also just in night now 10 to 11, just make a time. Okay, 10 to 11, 10 to 12, just 1 hour or 2 hour. I'll read this thing, because if you see these pathfinders and these books, now when I used to study, in whole one day I used to complete only 2 pages. It was difficult for me. But if now if we see this situation, in 1 hour we are completing one topic, so that makes a huge difference. You just have to do only one thing. Just if you're writing the key points now, just go through those key points. That's it. And please, I request, please buy Pathfinder. Please. Okay. So I'm done with today. Meet you next week.
Okay, ma'am. Thank you. So, students, if you
Sir, I'm
understood today's
finished.
stuff, please. Yeah, yes, ma'am. Please, yes, in the comment box. If you understood today's stuff, please comment yes in the comment box. Okay. Okay. Thank you. Thank you, everyone. And tomorrow we will organize one physics class from Diksha Pandey ma'am. Maybe we are organized. Let you know in a group. Okay. Okay. Now you can
Sir
leave the
is
meeting.
there?
Thank you, everyone.
Hello?
Sorry,
Sir
ma'am.
is there or not?
Yes, ma'am. Hello.
I don't know, sir is there or not. I think we should leave the meeting.
Yes, ma'am. Yes, ma'am.